From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on May 15th, 2025. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 92nd episode of Matters Microbial. As always, I am so very grateful for your listens, views, and comments. Means a lot to me. First, I have a new 3D microbial centrism pen I found on Etsy, as you can see here, plus I'm wearing it. I've added it to my collection. It's time for the summer before my last class, my beloved microbiology at the University of Puget Sound. So I'm thinking hard about what kinds of microbial shock and awe experiences I can bring to my undergraduate students. Remember, they only get one microbiology course here. I enjoy teaching my students how to isolate a specific colony from a mixed plate using these E. coli strains containing pigments, the genes of which were made by Sebastian Kosioba, as you can see here. They're very pretty. We sometimes call them forbidden Skittles. But even then, look at these strange colonies we see from time to time. Do they arise from two original cells close to one another with different plasmids, or is something weird and wonderful taking place? I always have my students look. I will continue to urge my students to really look at their plates, as when one of my students found what is very clearly Pentabacillus on a plate. The look on her face when I showed her photographs from the literature of this genus was just wonderful. And I will do my best to encourage creativity, as when I ask my students to create microbial memes. Like this one, made from a photograph of a colony of Myxococcus xanthus consuming a colony of E. coli. The student title, "There is no I in team or in Myxococcus xanthus," was just perfect. Creativity matters, friends. For today's topic, we return to my first microbial love, marine microbiology. I ask my students every year to take seawater samples and plate out dilutions, as seen here. Look at this complicated plate with auger pitters, pigmented colonies, gliders, and even a fluorescent colony that I suspect is due to siderophore production. Diversity is the rule in the microbial world. Years ago, a marine microbiologist friend of mine decorated an 8-ounce styrofoam coffee cup, stuck it to a deep-sea submersible, and let it go. As you can see, the pressure drove the air in the styrofoam out and made a tiny cup for me, which I treasure. In several prior episodes of Matters Microbial, we have chatted with microbial scientists about metagenomics, extremophiles, evolution, and life in the deep ocean. So I am delighted to welcome Dr. Valerie DeAnda Torres of the University of Florida to the podcast to discuss the work of her research team in decoding the metabolism of extremophilic microbes in the ocean and promoting women in science. Welcome to the Quality Quorum, Valerie. It's been almost a year since we met at ASM Micromed in Atlanta, and I'm pleased you could join us today.
Thank you so much, Mark, for the invitation. I'm really happy to be here.
I think you have a horrifying photograph of our first meeting.
It was amazing.
Well, I have a perfect face for radio. Let's put it that way. You know, Val, first, and you sent me so much interesting material, which will end up in show notes. But would you care to take a moment right off the top to tell listeners and viewers what it means to be a National Geographic Explorer?
Yeah, for sure. So basically, explorers are individuals from different backgrounds who receive funding from the society, from the National Geographic Society, to kind of protect and illuminate our world. So basically, being a National Geographic explorer means joining a community of scientists, educators, storytellers, adventurers that not just study the planet, but we act to protect it. And it basically means that the society funds our projects and equip us with training and networking and all the tools in order to kind of detect a gap in our world and kind of act to kind of protect it and to bridge this gap with compelling stories, evidence, science. Um, for me, it means to kind of highlight the importance of microbes in the planet and turn these discoveries into visuals and outreach that inspire others to care and to act. And we turn these discoveries into solutions that inspire the global community and basically to build a more sustainable future.
This is perfect because it's all about microbial PR. And much as I'm bitterly jealous that I've never given a TED Talk, that only is a little bit of PR. You're actually walking, swimming, striding, diving the
That,
microbial walk.
yeah,
So my point is this is really pretty amazing. And I want to say that you have this interesting background where you not only go out to sea on cruises to collect things, and then you come back and do the wonderful work in the laboratory, and then you do all the computational work. So you're an excellent person to talk about this new view of environmental microbiology. Um, so many different things. Do you want to give a thumbnail sketch of the sort of things that you do?
yeah, for sure. So basically, I've been working and kind of combining field work, lab work, and computational work for more than a decade. I did my PhD in Mexico and in Spain, and I've kind of always tried to kind of capture the microbial world in real time. And at that time, it was kind of how do we, in, just to put it in kind of an easy way, is kind of thinking about a metal detector, but instead of metal, it detects microbes. So
Yeah.
I started thinking about kind of ways to capture the microbial world, but microbes are the most diverse and abundant form of life in the planet. And so how do you do this? And most of them, we can't culture them in the lab. And most of them are unknown, novel lineages of the tree of life that we didn't even know that exist. So I've been kind of thinking about combining these three disciplines that I really love, and in order to find ways to capture microbial microbes and their metabolism and what they're doing in the environment in real time, and kind of implementing this in on board of ships and in the ocean in order to identify them. But not only that, but also what are those microbes telling us? We can create ways to protect the ocean, contaminated places, or ways in which we can identify things that are not working well in the dynamics of the ocean. And so it's kind of a circle that combines these disciplines in order to inform, to protect our planet and our oceans.
No, I mean, I have a saying, first evolved, last extinct, about the microbial world, and it's really true. One of the things that listeners and viewers need to remember is that we have a prejudice a little bit in the microbial sciences about growing things in the laboratory, but the hand of Darwin is on all that lives. And so
Yeah,
the process of growing things in the lab changes them. Evolution is taking place.
yeah,
So even if you have something that I've isolated from seawater, it may not be exactly the same way it was originally. And so the
exactly.
power of what you're saying is you're looking at them in situ, and not just in situ, but also in relationship to the other organisms that are there.
Exactly. That's super important. When we isolate them, we are only examine one part, one aspect, but we're in in nature and we can kind of see what is happening. It's not the same studying microbes in the lab that actually doing it in their environment.
So I was talking to someone on an earlier episode of this podcast about the things that I hear on it, like what you're talking about today. And I have this mixture of different feelings. There needs to be a German word for it, I suspect, where you're happy and jealous at the same time. So it's a
Yeah.
wonderful time to be a microbial scientist is my real point.
Yeah. Yeah.
So I wanted to also say that many people that we've talked to, or many people have the idea that you work at a lab bench, and you do, but you're also doing things on a ship.
Yeah. Yeah.
And sometimes that's just a matter of isolating and freezing things, taking back. And other times you're doing things on the ship to prepare things. I know this.
Yeah.
So it's like I said, so many different levels to what you're talking about.
Yeah. Yeah, I think it's very important. I was talking to my student about this. The way you work in the lab, it's very focused on everything on under control, conditions. On a boat, everything is different, right? And even when you're actually extracting DNA from this, from sediments, from the water, you have to consider that you're in a moving ship, right? So not only that, but also the environment, the community. Like you have to rely on having a community that is supportive and that is empathetic and that understands what you're going through, because you sometimes have 24/7 shifts. Like you're not like sleep deprived, you're working all the time. You have to, take into consideration other people that are working with you, that are working in the same ship. So it's more like a community in order to make things happen and make things work, which is very different from the lab. um and I also want to mention, like the last crew we went to, I was invited by this amazing scientist that I really admire, Mandy Joy. It was a female-dominated crew. It was a female-dominated crew, which was very unusual. And this really made things different in terms of how you show um, support and understanding and collaboration, and it was just amazing. But yeah, the dynamics not only at the um, lab level but also in the community level, everything is different when you're doing field work in the ocean.
It's not quite the same, but it reminds me of scientists that work in Antarctica because you really can't get away from the other people when you're on a ship. Now, true, you're there for weeks to a month, something like
Mhm.
that, and you're down in Antarctica for 6 months. And sometimes people get a little unusual in this. So putting kind of the emphasis on learning how to form a community is
Yeah.
also kind of a metaphor for the microbial world when you think about it.
Yeah, exactly, exactly. You have to, it's not about, it's about building a community that will allow you to survive during the month or 6 months that you are in nature, in my case, in oceanographic expeditions, but also doing field work in deserts. I did my PhD in the Chihuahuan Desert in Cuatro Ciénegas, and it's also an extreme environment. It's also very hot. It's very complicated to get there. Like you can get hurt. Like you have to rely on other people in order to survive in a some sort of way. So yeah, it's like the microbial world.
You know, you mentioned this. I had a student years ago who was doing hikes through northern Mexico, and he came to some of these anoxic lakes. And I don't know how well you can see this, but this is
Oh
a rock that's covered with what I think are tiny manganese nodules.
yeah,
And I,
yeah,
I haven't ever like looked at them under isotopic analysis, but it gives you the idea of you go to where the cool work is.
yeah, yeah,
And he actually said to me, "I'm really mad at you, doc, because you told me I would always think about microbiology, and that was my first thought when I saw this."
yeah.
Mission accomplished.
Mission accomplished. Exactly.
So one thing that I'll bet readers, listeners and viewers would be interested in hearing about is an example of how you actually went onto a submersible, which brings us to Alvin. And the reason why is you have to collect the material to process.
Yes. Yeah, I think going in the Alvin has been one of the most exciting experiences in my life. It was like the opportunity to go down to the bottom of the ocean. It's pretty much like you're in a spaceship, like you're in a completely alien world. Like you don't know what you're gonna find. And it was very amazing. Like the first time I went and I went down, um, I remember I was just like looking at the window, every little single creature, like enjoying, like going down, enjoying every single moment and absorbing everything. Uh, and I kind of felt like it sounds cheesy, but I felt like Gamora from Guardians of the Galaxy on a spaceship, like. And I was just like ready to go to the other side and collect samples. And you're like looking at the microbial mats, you're looking at the hydrothermal vents, you're looking at the jellyfish and the fish, and then it's just like a completely different world. And then it kind of changes the perspective of, okay, I'm in the bottom of at the bottom of the ocean collecting the sediments, and I'm going up. I'm processing these samples. I'm extracting the DNA of the samples, and I'm like looking at what potential microbes are living there, like just several hours later. Like it's just
Yes.
amazing that process of going and doing like kind of real time metagenomics. It was something that I've been wanting to do like for 10 years now. And like I was working on kind of implementing algorithms and computational pipelines to accelerate the process of discovering and understanding microbial metabolism. And now I can get to like do it in real time. It's just like it's very exciting.
So let's take a closer look for just a second. Okay, so we have a photograph of you in the hatch of Alvin, which you can see is a narrow squeeze.
Yes.
And then we have another photograph of you, not at the console necessarily, but certainly around some instrumentation. How many people are in the Alvin with you?
There's three people, the pilot and two scientists. And so, and the one that I'm looking, that I'm like kind of giving some sort of instructions is when we connect to the main ship, to the Atlantis, and we tell the crew how many sediment cores we collected, how many Niskin bottles, what we're gonna do next. We're trying to keep in touch with the main ship just to kind of tell them what are we going to do next because we don't know what we can find. On the second dive that I did, the current was very strong, so it took us away from the map that we had loaded. So in those kind of conditions, you just have to constantly communicate. um but yeah, three people. It gets very cold. That's why I'm wearing a hat and a blanket, and because I get very cold.
But is it not very hot when you get in?
No. No,
Oh,
no, no, no,
okay.
no, no, no. No, it's not. Um,
I guess it depends on where you
it
dive from.
it
Yeah.
it really depends. I mean, it's hot because like we go in the sphere around 7:30, 8 p.m. So it's not really hot. Yeah,
So I'm gonna put a link to the history of the Alvin. And I was fortunate enough to see it at Woods Hole when I took the Marine Biological Laboratories microbial diversity course. Could see the scorch marks on some
yeah,
of it from
yeah,
the first time they saw the hydrothermal vents.
yeah.
Saw the hatch and realized there was no way I could fit into the device, but that's okay. That's okay because I'm a touch claustrophobic, so I'm feeling exactly how I felt when I was talking to Hazel Barton about cave microbiology. I'm glad other people are doing it. Um, but let's make sure listeners and viewers know you were taking core samples and water samples, and they're on the outside of the Alvin. Why were you taking those samples in that specific location?
Why? So we were at the Gulf of California, or Mar de Cortés, Sea of Cortés, and this place is very unique. It kind of marks all the conditions for a unique environment. It's a very um, unique place in terms of has high sedimentation rates, hydrocarbon production. It creates a dynamic in which communities are thriving, microbial communities are thriving, and it's very unique in terms of their diversity. Every time we sequence, every time we analyze those microbial communities, the more microbes we discovered. So this was part of an NSF funded grant led by Mandy Joy, Karthik Anandramar. He's gonna kill me if I don't pronounce his last name correct. Karthik and Roland's lab. So we were very excited, and I'm very thankful that I was invited along with Brett Baker. We were invited to this oceanographic expedition, and it was amazing.
I just met Brett when I was at University of Texas Austin, who speaks very highly of you. I met his dog, who was the sweetest dog ever. It was like I said, you have these, and I know Roland Hotzenpiekler quite well.
Hudson Pickler.
Yeah. You know what's helpful about him is in his SIG file and his emails, there's a link that tells you how to pronounce it.
How to pronounce it? Yeah, Karthik should do the same.
I think it's a good idea. In any event, now this is interesting because, so many people have the idea that the bottom of the ocean, and I remember when people thought this, was dead. And of course it's not.
It's
First
not.
evolved, last extinct. But not only that, with certain forms of these microbial communities, they can support fish, invertebrates, all kinds of things as well. That's the whole business of what happens when you're looking at a hydrothermal vent. It's this like hotspot of diversity.
Exactly. Yeah, I mean, funny story. Like when the first analysis of the first dive to the hydrothermal vent systems, I think it was in the 70s.
It was.
They they didn't send biologists, only chemists or
That's right.
geophysicists. Like they didn't think that there was going to be life down there.
And,
And
and they
then
and they didn't
surprisingly,
and they didn't think to turn off the lights. That's the part I thought was interesting.
yeah. Yeah, exactly. So here we are years later analyzing and still discovering and still like we don't know, like one of the least known ecosystems on the planet is the deep ocean, and we're starting to understand and analyze it, and it's just amazing.
One of the themes that we've talked about on this podcast many times, and it's part of my classroom curriculum, is we get very reductionist. We look at one organism by itself. And of course, that's not how any organism really lives. And so the fact that you're looking at the communities is so important. And you see this amazing diversity. And it's interesting because, we all love the archaea. We do. They're interesting and all the rest, but there are lots of bacteria down there as well doing their
Yeah,
thing. And
yeah,
sometimes interactions between those two,
yeah,
which I find endlessly fascinating. So that's you took the samples and then they have to be carried back up.
yeah. They have to be carried back up. I have a picture of it with me and Maggie, who's a PhD student at Karthik's lab, taking those sediment cores back to the cold room. We have to bring them at a cold 4 degrees in order to slow down the metabolism, in order to process them. So you have to, protect yourself, like have the proper gear because you spend a lot of time in the cold room. So now you have to measure the cores, look at everything, all the metadata that you can collect for every single core. It's very important. Presence of hydrocarbons, presence of, the different smells, like how every sediment is different, every site is different, the geochemistry is different. So we collect and analyze all the course, and depending on what we're gonna do with them, if we're gonna use them for enrichment. So for example, Roland's lab was doing enrichment of methanogens, or Karthik analyzing the viral community. They were analyzing the viruses and the plumes around the water column. So depending on what type of post processing you're gonna be doing, it's kind of the steps. But bottom line, you have to bring them back to the cold room, and then after that, you decide what to do with each one of the cores or which one of the water samples.
It's like the old saying from ancient zoo work, you got to bring them back alive.
Yeah, exactly. That's the whole point.
So one of the things that you've said you're most interested in is the kinds of metabolic pathways and how
Mhm.
they differ in different environments.
Yeah.
So you should probably tell our listeners and viewers, I mean, when we talk about metabolic pathways, many of us learned about glycolysis and Krebs cycle and electron transport, things of that nature. It's part of what I call our eukaryocentric view, which is
Exactly.
a problem, right?
Yeah.
What's more diverse and various than the microbial world?
Yeah, exactly. So I think I like to put it in a way that, as you mentioned, the eukaryote centrism, as eukaryotes, humans, plants, animals are very boring metabolically speaking.
It's true though.
We only do a respiration, oxygen, sometimes a anaerobic respiration, but respiration, um, and photosynthesis, which is the other way around, basically. So, in that aspect, when we look at the microbial world and we see how many electron acceptors and electron donors and all the things that they can eat and they can process, is just like a whole different universe. And that microbial metabolism is just astonishing. How all the ways, all the things that they can breathe, they can use, they can. It, I'm very passionate about microbial metabolism. Uh, but yeah, basically exploring the ways in which organisms conserve energy,
Yes.
and the way they do it in the bottom of the ocean, it's pretty special. When we think about photosynthesis, we have in mind, plants and surface ocean. But what happens in the bottom of the ocean is that even though even we have no light, microbes can use inorganic compounds, all the things that are coming from the earth crust and minerals, in order to, use them, consume them, and conserve energy. So yeah, that's the things I'm very passionate about.
Well, I mean, one of the things that's been really, and we've talked about on the podcast before that I just love is when very hot water around a hydrothermal vent hits cold water, a small amount of light is generated, this
Hmm.
form of triboluminescence, and there are some cyanobacteria that can harvest that light, which is just amazing to me because that could happen on other planets,
Exactly,
if you're into
exactly.
that.
And
But it's funny you talked about eukaryocentrism, and I'll add a second one, which is oxycentrism, because as you say, this business, and I showed that in the little rock with all the nodules on it. And in one redox state, it's soluble, and then when you steal or donate an electron to that compound, that is what respiration is, and it precipitates. And there are people that used to use that to help with mining or
exactly,
to reduce toxic compounds. So we can learn from the microbes, is my point. Um,
exactly.
by the way, the third one, and I know you'll appreciate it, is what I call colicentrism, which means microbiologists fall into this all the time. They think everything does its lifestyle the way E. coli does. That's not true.
And it's not true. Yeah, actually, is one of our problems in terms of analyzing microbes that haven't been cultured. They don't exist in databases. There's no reference in the data sets, and that is it is a problem in the sense that it takes us a long time in order to decode those processes because we're trying to compare with what we know, right? But since that is something that we don't know, we have this big gap of knowledge.
Right.
We have to be creative and find other ways, computational ways, in order to infer, in order to identify these fingerprints and try to make sense of what is happening. And you have to use, thermodynamics, the community, whether or not that process is, you know, potentially happening, because there's nothing that you can use to compare with and you can't grow them in the lab. So you have to be creative in order to kind of have an idea or a hypothesis of what those microbes are doing in that environment.
And you really have to take off our blinders about life as we know it. Because I remember when I was, you got to forgive me, Val, I was this microbial nerd as a boy. And even though my eyes are not great, I could still do some microscopy. And I remember looking at some anaerobic sediment. I was like 10 years old, and things are just moving around like crazy. And I remember thinking, but without oxygen, they're not making very much ATP.
Yeah.
But that doesn't matter, does it?
No. No.
And see, but you think it does. That's the issue that's so
That's
interesting
the
to me.
that's the problem. Yeah, people think, well, there's no oxygen. What are they gonna do? How are they gonna survive? Well, the microbial world can use other things that are not necessarily oxygen
And do very well. That's the part
and
I
do
thought was
very
so
well.
interesting.
Yeah,
Right.
and do
S-
very, very well.
So I know that you've done some work with, I think, methanogenesis. And the issue there for listeners and viewers is that methane is definitely a greenhouse gas, about 40 times more pernicious, would be the word I'd use, than CO2.
Mhm. Mhm.
And so that should bring everyone's attention up to the value of looking at these things.
Yeah. So basically, when we found this new genomes that when you put them in the tree of life, they don't look like anything that we have seen before. And then you start looking at what is the potential of these microbes, right? We name them after Thomas Brock for his great work on microbiology and hot springs in Yellowstone National
Mhm.
Park. Actually, that was the first book I bought when I had the means to purchase a book. That was the first book because I was totally amazed. Yeah, a giant
Yeah.
rock microbiology. So it was it was very difficult to pinpoint the potential of these microbes because they look like methanogens. However, they didn't have the markers or the fingerprints for methane production, which is MCR. Um, it was it took us a long time because they were living in the same kind of environment. They were using the same substrates as other methanogens. The other archaea that were known to have this um, kind of machinery were methanogens, and so at the end, we propose a way in which these organisms that are lacking completely the methane production machinery are living along with methanogens, consuming the same substrates, but not producing methane. And this has huge implications for climate change or for global models because we are so at isotopic analysis and from geochemistry and other studies, we're pinpointing the presence of organisms that were uh, using similar substrates but not producing methane. And so far the only archaea known to use methylated compounds were methanogens. Then we discovered this prokaryotic genomes, and after that, a hu- several studies have also identified this potential mechanism in other archaea. So it's not like an isolated process; it's something that is common in nature, and that can help us kind of understand the production and consumption of methane, and obviously to understand our climate, because at the end of the day, the bottom of the ocean is kind of the engine that drives the biogeochemical cycles in the planet, even the atmosphere and in our planet. So it's very important to understand what's happening down there, because it's the largest reservoir of carbon in the planet, the marine the marine sediments, and this reservoir of carbon is in the form of methane. So we have to think about what would happen if we release that methane, right?
Bad
So
things.
yeah, yeah,
It reminds me, it's not the same thing at all, but a lot of ruminants, cows and similar ruminant mammals, will generate quite a bit of methane from their digestive process, turning
yeah,
hydrogen gas and CO2 made
yeah,
from fermentation into methane for energy.
yeah.
Um, Turns out kangaroos are ruminants, but they don't make methane. And so this is kind of like that's the mammal terrestrial version of what you're talking about.
Exactly. Yeah, exactly. And understanding why is this, and understanding kind of the implications, and what are we going to do with this information, how are we going to protect our oceans and our planet, which is kind of what I am very, very interested about.
Val, does National Geographic get any federal funding? I'm kind of curious about that. And also, I gingerly step into this. Has any of your funding been threatened by recent cuts that we've seen in the budget? And your thoughts on that?
So the National Geographic is a private foundation, the society, and it's the grant that I got is both supported by National Geographic and the Smith Ocean Institute partnership. Um, so this partnership is not a federal funding. So for this, I'm not concern in terms of federal funding. Obviously, as a scientist in the US, I am very concerned. I'm very affected, as all are, um, by the nature in which all these changes are happening and affect the science that we do. So, for example, if this would have happened, if these cuts would have happened last year, I wouldn't be able to do the oceanographic expedition that I did, that was led by Mandy Jo. It was an NSF funded, a federal funded expedition. So there are many expeditions, there are many grants that are being affected. Um, for in the short term, let's say, for this specific Falcor cruise that I'm gonna be doing in November, and the chief scientist is Brett Baker, and I'm a co-PI with Alison Santoro and Cecilia Alonso. It's not threatened by federal funding, but obviously, as a scientist doing research in the US, as a Mexican, as a woman, it's definitely concerning. Yeah.
I appreciate your thoughts. So, would you mind like throwing an experiment or an avenue of research that you were really excited? I think we have time for two of them that you could kind of tell us about, because there's nothing that makes me happier than seeing folks being excited about their work.
Oh, I'm excited about a lot of things, but since we only have time for two, um, let's see. I am working with this group of amazing scientists from UT. Um, we are trying to understand the degradation of PFAS compounds, microbes that can degrade contaminants, the forever chemical contaminants, and try to understand, um, if microbes can degrade this, one of the most toxic chemicals that exist on the planet. The This is one of them that I'm currently working on with my student Swati, and implementing real-time metagenomics on board of the Falcor, not only for kind of the discovery aspect, but also one of the things that I wanted to do during my PhD was kind of gather all the evidence for conservation purposes.
Hmm.
At the end, we didn't, it took us, it was kind of late in the sense that despite all the efforts to conserve this natural protected area, the lagoon where I did my PhD no longer exists because, yeah, because the deep aquifer that was supporting all the ponds and lakes got no for agricultural purposes.
Got
So
diverted. Yeah.
they were using that water in order to um yeah. So it was very sad. Now that place that hold stromatolites and microbial mats and like the oldest evidence of life on Earth, it doesn't longer exist. Just a few kind of ponds remain, but it's kind of um if a reflection of what's happening in the planet. We're consuming more than what this planet can hold. And so, how we can use that information, especially microbial communities, are not used for implementing conservation strategies and policymakers, how we can, especially when microbes are the ones are the heart of every ecosystem on the planet, including ourselves. So why not using microbial communities as an indicator of what of ecosystem health, right?
Yes. Yes.
So that's kind of what I kind of want to do in order to find ways in which we can do this in real time, identify microbial blueprints that can indicate contamination or this ecosystem is degraded. What can we do in order to protect it? I mean, microbes are going to survive without us, but we can survive without them. So,
Absolutely. Yeah.
yeah, that's one of the things I'm excited about
I,
doing
I
in my research.
We recently had Rosie Algado on the podcast, and one of the things that most interests her, she and her research group, is the ability of restoring some of the fish ponds that the indigenous Hawaiians had set up, and they had a whole structure to this. And it's not the same thing as what you're talking about because they're living in well, they're living in concert with it as
Yeah.
opposed to piping away all the water. But the
Mhm.
point is, you can get some idea of what's going on as you watch how communities change.
Yeah.
I run into people fairly often, and I know you do too, who will say things like, well, it's just germs. Boy, that gets me going.
Yeah.
Or it's just bugs
Yeah.
or whatever. But the fact is, microbes are the base of the biosphere itself.
Yeah.
And so you're not saying don't do X or don't do Y. What you're saying is, what is the effect of doing X or Y, which is a really important question.
Yeah.
So, congratulations on that.
Thank you.
Even though I'm sorry the lagoon's gone.
Yeah, same. Very sad. Very, very sad.
So that's one thing. And by the way, uh, just to make you laugh a little bit, you were drinking out of your water bottle a moment ago, and I have my
Yeah.
students look what kind of microbes live in there. And you'll be glad to know that in plastic bottles, I keep finding methylobacteria. And I'm pretty sure that they're living off something being like leached out from I bet if I did SEM, there'd be little pits.
Yeah.
Right. I don't see them in metal, but isn't that fun?
It's very now I'm gonna be more conscious about it.
It's okay because I'm going to send you a sticker of what we call our water bottle buddies. Those are the organisms that live there. There you go.
Yeah, I love that. I love that.
So you had a second experiment that or a avenue of research that really excites you.
Yeah, so one of the things I'm very excited about is using current microbes in order to as a fossil record of early life.
Oh, yes.
Yeah, so I was very interested in kind of the the oldest evidence of life on the planet and what were the first microbes that appear. So one of the things that I have been doing, I've been working on is
Okay.
using microbial genomes in order to reconstruct the ancestors of those genomes. And so it's like, imagine we can't have fossils, like we don't have the bones. We can, build a dinosaur. We can't really know how those ancestors look like, but we can use computational algorithms in order to model what could happen in the past. So that's one of the things that I'm very excited about. And with this, I was very well, and was very excited to be part of this um, big project and big analysis that allow us to reconstruct the oldest ancestor of Asgard archaea. So basically, the archaea that gave rise to complex life. So I spent a lot of time working on modeling the metabolism of the ancestors. It was just like looking into the past and model what could this ancestor that gave rise to complex life look like. And I remember I was um, on the computer looking at the genes and looking at the presence of a specific genes, and like putting those kind of points on in my head and making sense of all of this. And every day was like, oh my god, they could have this. Oh my god, they could have a full electron transport chain. Oh my god, they could have. And it was it was very exciting, and this was during the pandemic that I was doing this work. And um I remember I was like, if I was an archaeologist, this is like, if you're going into a pyramid and you're doing this discoveries and you can't see and you can, or a paleontologist, but instead, instead you're looking at your computer, but it's like everything is kind of in your head and like making sense of all of this. You can't really go to the field and try to figure it out what the ancestor looked like, but you do it in your computer. It's not as
Mhm.
exciting, but it's one of the things that I'm very excited about.
No, no, no. You're looking for echoes of the ancestors,
Yeah,
right?
yeah,
And they're there. I have a former undergraduate research student. You may know her name, Paula Welander at Stanford.
yeah,
And she was one of my first research. I have pictures of her when she was like 18 working in my lab. Looks pretty much the same. She's one of
yeah,
those people,
yeah,
but looks
yeah.
again at something that can be preserved in the fossil record, these hopanoid molecules
Exactly.
in particular.
Yeah,
And what's important to let everyone know is that you're looking for echoes.
mhm,
Very seldom do we find something that enables us to say what was taking place in evolutionary time.
yeah,
And it's funny because I make evolution happen with the bacteria that I study in a week's time.
yeah,
And I think that Von Cooper has a wonderful outreach program in this area. But the point is to go back to the first ancestors.
yeah.
And I don't know if you feel the way I do, and I'm not disrespectful, but the people that spent like years and years getting them to grow in the lab, are they really the same as they were?
Yeah, it's
But if
funny.
but if you don't have them growing, how do you study them very well other
Exactly.
than what you're doing?
Exactly. Yeah, it's funny that you mentioned Paula. We have a collaboration, an amazing collaboration that was one of the most exciting projects that I've been involved. We were actually looking at the presence of hopanoids in Asgard archaea because they're present in the fossil record, right? So
Mhm.
when a microbial community dies, what remains are triterpenoids, so it's lipids that are in the membrane, cholesterol, ergosterol. All of these molecules are recalcitrant and they can survive. Persist in the fossil record for billions of years. So paleontologists go to the fossil record and extract these molecules, and then geobiologists like Paula identify current microbes that could, produce these molecules, hopanoids or esterols. So we were wondering whether Asgariarchaea, since they are, the oldest prokaryotic ancestors, produce hopanoids? Well, it turns out that after doing a lot of bioinformatic analysis and with the amazing PhD student Hanun Makshi that now is doing a postdoc at University of San Francisco, we spent a lot of time trying to figure it out whether they could produce hopanoids. We identified that they produce diterpenoids. And this has something that nobody has seen in the entire archaea domain of life. Nobody knew that diterpenoids that were only found in plants were present in Asgard archaea. And turns out that paleontologists haven't even looked at the presence of diterpenoids. So now we can tell them, hey, look for these molecules because they could be present in the fossil record. So it's kind of like going back in circle and like combining one of my passions, that is paleontology, geobiology, computational, and lab work. Hannon spent 2 years trying to clone the genes in order to see whether they were functional or produce this lipids. The only microbe that produces this type of diterpenoids is a Microbacterium tuberculosis, and it uses as a defense mechanism for when the white cell tries to kill the
Mhm.
bacteria. So it's a kind of a defense mechanism that allows Microbacterium to survive. So it's very exciting because we haven't even seen this type of. Now we have more avenues to explore and more open questions that, yeah, it's just very, very exciting. But yeah, it's going back in circles, and it's one of the my other passions is using microbial metabolism as a fossil record of early life.
I have to say, the first thing that I tell every one of my research students is the microbial world is smaller than you think. And people think I'm making a joke, but what I mean is it's really important to be kind to everyone because there are all these connections um,
Yeah,
everywhere. And
yeah,
so, I can't speak for you, but when I was an uninvolved person in my 20s, I was not a pleasant individual. I have it on good authority. But you eventually learn that it's better to be collaborative and it's
yeah,
better to be kind
yeah,
and to reach out and chat with people. And
yeah,
and that's what I want everyone to take away from this kind of discussion with you
yeah,
is how collaborative, how kind,
yeah,
how interactive the way
yeah,
that science is being done. Are there
yeah,
exceptions? Sure.
yeah.
But more to the point, I have had so many good experiences myself in microbiology, and it warms my microbial heart to hear about yours.
Yeah, yeah, definitely. That's something that I always encourage my students and my friends and people because, there's a lot of stories out there and people that are not nice, that are not kind.
Mhm.
At the end of the day, you have to collaborate and you have to
Yeah.
learn that science is a collaborative effort. And yeah, it's very important to have this take home message that it's very important.
Well,
Collaboration is crucial.
you've probably seen the old-fashioned thing where they ask school children to draw a scientist, and for many years they drew, dudes, right?
Yeah, yeah,
And now it's starting to change a little bit, and that's why it's important to let people know that science is a human profession, and it has
yeah,
all the foibles of one,
yeah,
but it also has the hopeful one. And I have an expression, and I wish I could find a good cartoon for this. A big tent is where you say that you have a bunch of different people involved in a common cause. It's a big tent issue.
yeah,
We need a big petri dish.
yeah,
And that's what I have found. If you ask people for their opinion or their advice or help, sure, there are some people who don't have time for you,
yeah,
but there are a lot of people who do.
yeah,
And
yeah,
your whole career has been kind of an illustration of that.
yeah, yeah. I think I learned very early in my career. I mean, as a Mexican scientist, I faced many challenges throughout my career for many years. And the fact that I didn't grow up having or knowing what a scientist looked like, as you mentioned,
Mhm.
or having female role models, and not having the experience of, not being treated in the same way as my male friends just because I was women, and I was meant to do other things like going in the kitchen and, because it's a very patriarchy culture,
Hmm.
that sometimes you just have to fight. And so my entire life has been fighting against this patriarchy model in which you can show that you can be a scientist and you can pursue your dreams that not necessarily are the standards of what the society expects from you. And I've learned that during also my scientific career, like there's going to be a lot of people that treat you in the way that you don't deserve. Like I've been told, you're never going to be a scientist. I've been told, oh, you're not bad, you suck. Or you have been told many other things that I'm not going to mention. Um, so that happened throughout like many years in my career, in my life, until I found amazing people, amazing collaborators, amazing scientists that not only support me and help me and provided me with, a safe environment in which I could, develop all of these ideas, and which I'm very, very grateful. And I could name all of them. So it's part of also feel that you are in a safe environment so you can, uh, you know, do all of the things that you want to do. And it is very important for the next generation of scientists and trying to provide and like break down those barriers for them to feel that they are in a safe environment where they can, thrive and
Yeah.
develop all of these ideas, especially for women, especially for minorities, and like to see themselves reflected in real life scientists like myself and like Mexican scientists like my dear friend Giovanni Santiago. Um, so yeah, it's very important. Diversity and collaboration and empathy, provide safe environment in science is crucial.
Diversity and collaboration is the microbial world when you think
Yeah,
about it.
yeah,
And
exactly.
you've met my wife, Dr. Jennifer Quinn, who's a mathematician, and she tells very similar stories
Yeah,
about expectations that people have, and I think that the outreach that you're doing is really something that a lot of people talk about it. You're doing something about it, is what
yeah,
my point is.
yeah, yeah, exactly. Yeah, it's very important. It's very important to do outreach, especially reach out to children, especially to reach out. I've been giving lectures at middle schools, high schools, undergrad level, like I wish like I do this for my younger self, and I wish I could have, a real life scientist like myself. And I get emotional because that's something that I always wanted to
Hmm.
do, right? Like always wanted to have this kind of interaction with somebody who, was just curious about nature, exploring nature, and like doing it as a profession. So it's very important not just to explore, but also to connect with the next generation of scientists. And, diversity starts by awakening these children and tell them you can also, if you're interested, you can also do it. Like there is a
Hmm.
profession for you. You can be a scientist if you want to. So yeah, it's very important to do this outreach. And yeah, it's part of, I feel like sometimes when I get frustrated and you know science wise, or the work is not um happening in the way that you want, but then you have, you know students from different places and telling you, I saw your talk and it's very exciting. And how can I do this? And this is when you realize it goes beyond your research, it goes beyond publishing a paper. It has
Hmm.
implications. When I give talks to in Spanish to students from Latin America, and then you see them sell, and then you see that they get excited and they get, they question, they have questions, and it's something that goes beyond the actual research. And yeah, I feel like it's super important, and I'm also very passionate about that.
Well, and the good news is that you've sent me all kinds of links. Your wonderful website has all this. I mean, I'm going to be linking to things like crazy in the show notes. No, I'm quite serious about this because I think it's important for people to see.
Yeah.
And I don't want to give you uncompensated work, as the saying goes, by saying that keep on doing the good things that you're doing. But what I also know is that you can't help yourself because I think you're trying to help younger you.
Yeah. Yeah.
And
Yeah,
and
it's one of the things that sometimes is like, why you keep doing what you're doing? It's like it just goes you beyond that.
yeah,
It's
and
yeah,
it's really true. I mean, I was this, raised in an area where nobody went to college, and my poor parents had no idea about this science thing. But I'd always wanted to do it. I'd always been interested in it, and they did their best to support me. But not everybody has that experience,
yeah.
and that's why the outreach that you do matters.
It's very important. When I go back to my elementary school and I see like how I was when I had that age, I was like all the things I've been through, and I just it's part of the things that
Yeah.
keeps me going.
Well, I got to tell you, this has been really a fabulous session. I mean, as you say, we could talk for hours more.
Yeah.
So the good news is we can do this again some other time if you like.
Yeah, for
Uh,
sure.
and and especially because what I'm looking for is someone to just really sit us down and talk about the Loki Archaeota.
Yeah,
Um, so you think about some people who are willing to do that, or maybe you will do that later.
yeah,
Um,
and the new culture from Horarkyoda, there's now three cultures of Asgardia.
ah,
So yeah, we can talk about that too.
I just love the idea so much. But I want to really thank you for your time. I am such an admirer of the different levels at which you do your work, from down at the bottom of the ocean to on the swaying deck of a ship to back at the lab bench to at the keyboard and then at an elementary school.
Yeah.
My question is there enough Val to go around?
I think so.
That's wonderful to hear because you're doing a really good service. So I want to
Thank
wish you
you.
the very best to you and yours, and I want to thank you again for this opportunity.
Thank you so much, Marcus. It was really fun. And yeah, thank you for inviting me.
Of course. I hope to see you at Microbe. Aren't you giving a
Yeah.
talk?
Yes, I will. We're gonna have a session talking about Asperger Kia and early metabolism. So yeah, Carolina
So
is gonna be the chair of that session.
you need to chat me up for some more guests, okay?
Yeah,
So think about that.
yeah, for sure. Awesome.
Thanks
Yeah.
thanks so much again.
Thank you so much, Mark.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode with epic links can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Valerie DeAnda Torres can be found on land in the Department of Microbiology and Cell Science at the University of Florida in Davie, South Florida. Dr. DeAnda Torres is also part of the Fort Lauderdale Research and Education Center and the National Geographic Explorer program. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our quality quorum for today. See you next time on Matters Microbial.